Hello my dear students, welcome back to Biology at Priyajit. Today we'll explore developmental biology, previously which was known as embryology. So how from a single zygote to a complete organism is formed. This journey is truly fascinating. What is the definition and what is the modern concept about developmental biology? Traditionally, it was known as embryology, that is a development from zygote to adult-like stage. Modern view is the developmental biology, which includes growth, repair, pattern maintenance, aging, and death. Development begins from germ cell formation and ends at death. Historical background: developmental biology is as told as biology itself. Early philosophers explained development without experiments. Scientific basis established after cell theory in 1839 by Matthias Jacob Sladen and Theodore Swan. There are two major theories regarding the initiation, regarding the belief of development. That is, the first one is the theory of preformation. Gamete contain a miniature adult or known as homunculus. So development is the growth plus unfolding. So two groups were existed at that time. Number one is ovulist, who stated that homunculus is found in ovum, and second group is spermist, which is also known as animalculist. According to them, homunculus is found in sperm. So key scientist is Antony van Leeuwenhoek. So their problem was infinite nesting of homunculi, that is illogical. Theory of epigenesis proposed by Aristotle. Development, gradual differentiation from uniform mass. There is no preformed organs, and it was supported by William Harvey and Caspar Friedrich Wolff. The famous quote was there, omni vivium ex ovo, that is, life comes from egg. And this modern biology accepts this epigenesis, that is, with modifications. So experimental embryology, later it was developed. The key contributors were Wilhelm Rox and Hans Spemann. Spemann discovered that organized region in embryo, and he awarded the Nobel Prize in 1935. Conclusion: development is epigenetic and cells initially intermediate. Basic mechanism of development. Development involves three major steps. First one is the growth, that is cell division plus synthesis. Interaction, that is cell movement, cell contact and induction. Differentiation, cells become specialized, which leads to morphogenesis or formation of body pattern. Cellular basis of development. Development is a coordinated cell behavior. The process includes cell division, differentiation, migration, pattern formation, morphogenesis, and cell death. Then the process is controlled by genes, which ultimately produce proteins, which mediate the key event. Gene regulation in development. All cells have same DNA, that is genomic equivalence. Differences arise due to differential gene expression, and gene control determines cell shape, cellular function, and behavior. Differentiation. What is differentiation? Differentiation is the process where cells become structurally and functionally specialized. That is from the stem cell, muscle cell, blood cell, nerve cells, cardiac cells, which are both structurally and functionally different from each other. So the formation of the different types of cells from a single type of cells is called as cellular differentiation. Example: neurons versus glial cells, which is controlled by gene expression and non-coding RNA. Cell fate and potency. Cell fate, that is what a cell normally becomes. Cell potency is the total potential types. Totipotent cell, that is there is all cell types. Pluripotent, multipotent, unipotent, etc. Potency decreases during development. So what is the commitment? Cells become restricted to a specific fate, that is commitment, and it happens in stages. Mechanism of commitment. Cytoplasmic determinants. Unequal distribution of molecules during division is the cytoplasmic determinants, and it leads to different cell flights. Induction, that is one cell influences another cell. The induction is of two types: instructive, that decides the fate, and permissive, which allow progression, and it requires competence, that is the ability to respond. Lateral inhibition. What is lateral inhibition? Cells inhibit neighbors from differentiating, and it creates regular patterns, which is important in neuron formation. Mosaic versus regulative development. Mosaic development, when the fate determined by internal factors and their autonomous specification, it is called mosaic development. And regulative development, when fate is determined by cell interactions and it is conditional specification. Most organisms show both types. Pattern formation, organization of cells in embryo. Cells know their position via positional information. Morphogen gradient. Morphogen are the substances with concentration dependent effects. Forms means gradient. It determines the cell fate. Segmentation and compartments. Body can be divided into two segments, into various segments. Examples: insects are vertebrate, somites, etc., and it is controlled by homeotic genes. These are the image showing the homeotic genes. Homeotic genes provide positional identity, encode transcriptional factors, and mutation of the homeotic genes causes wrong body structures. So what is the conclusion? Development is genetically controlled process, and it is a coordinated cellular process. It involves growth, differentiation, and pattern formation. Still an active research field, that is developmental biology. So if you found this class helpful, don't forget to like, subscribe my channel and share with your friends. See you in the next class. Thank you, my dear students.